Hey guys, so I am here to show you how to do this foldable. This is going to be what our finished product looks like. All I did was take our two papers here. I folded them on the dotted lines. Blah, 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 blah. Doesn't have to be perfect. So that is one that's folded. Here is another. You know that you're folding it right when the little words are at the bottom, and then this one just fits into this one like this. You can staple the top or tape the inside. For this one, I just color-coded the bottom so that you guys could see more easily the levels in the different pieces of paper. Okay, so let's go into the foldable itself. All right, so I have not stapled or taped mine yet. So all right, so take a moment to pause if you need to write this down. I am going to go through everything. So we have a couple of important facts about chloroplasts. Chloroplasts are found in leaves and other green parts of plants. There are photosynthetic bacteria, and there's algae that also have chloroplasts. Chloroplasts contain a green pigment called chlorophyll. So chlorophyll is a type of pigment. There's actually several different types of chlorophyll, and there are lots of different pigments. So we do need to know that pigments are molecules that reflect or absorb light. So chlorophyll absorbs all the colors, most of the colors of light except for green and yellow. They reflect the green and the yellow. The structure of the chloroplast is really important. Chloroplasts have two membranes. They are separated by a very narrow space. They also have a structure called thylakoids inside. These are flattened, hollow, interconnected discs or sacs. When stacked, these thylakoids form something called a granum. Several granum are grana as well. All right, so stroma is the fluid outside of the thylakoids. The stroma is like the cytoplasm of the chloroplast. It contains ribosomes, chloroplast DNA, and enzymes. So let's take a moment, and if you haven't already written that down, go ahead and pause the video. I'm going to move my picture up, and we'll do our labeling here on our chloroplast structure. All right, so I don't really like the shading of this picture a whole lot. I just created it for this video. So this white layer here, this is the outer chloroplast membrane. And then this, gosh, you can barely see it. Let's say, that's the space. I'm just going to trace around it. Sorry, guys. I didn't realize it was going to copy so bad. All right, so this is the inner chloroplast membrane. I'm going to go ahead and do a line here just so that it's more clear for you guys. It's not pretty. And then all of this space here is the intermembrane space, so the in-between. All right, so in the rest of the picture here, it's a little bit clearer. This individual sac thing or disc, this is a thylakoid. Remember when we have a stack of thylakoids, that's called a granum. So this whole thing is a granum. I am going to just make a little note that grana is several stacks. So this chloroplast has 1, 2, 3, 4, 5, 6 grana. These round shapes, these are, of course, our ribosomes. These two shapes here, I don't know if it looks like it to you, but this is chloroplast DNA. And then the empty space that is in there is our stroma. All right, so there is our chloroplast structure that is labeled. All right, so now we're going to flip into our light-dependent reaction. We're going to start with our facts and information. This is the most complicated of the two parts of photosynthesis. All right, so there's a lot of stuff going on here. So take a moment now to pause to write or listen and then pause to write. So the first thing that we need to know is we need to know that we're going to start with our reactants. Our reactants are light, water, a molecule called NADP+. So you'll remember hopefully in cellular respiration that we had a molecule called NAD+. So this one is very, very similar. And we're also going to use the reactant ADP, The products that we're going to make in this light-dependent reaction is oxygen, NADPH, so this is our electron carrier, and ATP. Both these two molecules are going to go on to the next step. And the oxygen is released to the chloroplast for the plant to release into the atmosphere. So I'm going to go through these 6 steps here, and then again as I annotate the drawing. So the first one, light is absorbed by photosystem 2 and captured by the chlorophyll. Remember, chlorophyll is the pigment. In step 2, we have a process called photolysis. So remember, lysis means splitting. Photo is light, so this is light splitting. This is actually light splitting water molecules. So water molecules are broken to release hydrogen ions, oxygen, and electrons. The electrons will then be captured by a molecule or protein called photosystem 2. The electrons are going to travel from photosystem 2 to photosystem 1 in step 3. Just like in the electron transport chain in cellular respiration, they are going to lose energy along the way. Also, just like cellular respiration, as the electrons are traveling, the hydrogen ions are going to be pumped into the lumen. In step 4, remember our electrons have gotten to photosystem 1 and they have lost their energy, so they're very, very tired. So the chlorophyll in photosystem 1 is going to capture light energy to re-energize the electrons. In step 5, those electrons are finally going to be passed to a final protein who's going to combine the electrons, with NADP+ and H+ hydrogen ions to form NADPH. And then in step 6, we now have a buildup of hydrogen ions in our lumen, or the inside hollow space of the thylakoid. So we have an ATP synthase that will be able to remove the hydrogen ion buildup from the lumen, and with that movement can then join ADP and a free phosphate ion to form ATP. I do want you to note that NADPH is an electron carrier. It's very, very similar to NADH, but you do need to remember which one is which, and most people pretend that the P And NAD, pH stands for photosynthesis. It doesn't, but we pretend. Okay. So let's annotate our drawing here. So this is our light dependent reaction. We'll move it down a little bit so that you may not be able to see the writing, but we can focus more on the image. So the first thing we actually need to do is, I'm going to use my green. We need to make sure that we understand what this is. So this thing here, this is our thylakoid. Okay, so that means that this space out here, this would be outside of our stack. So this is our stroma of our chloroplast. And then this space in here, this is the lumen, or the space inside a tube or sac. So this is a sac. So this is the space inside of our thylakoid sac. So it is a lumen. This guy here, this guy is photosystem 2. Oh geez, I cannot spell. Photosystem 2. Sometimes he is referred to as PS2. This is photosystem 1. Or PS1. And then this guy down here is our ATP synthase. So I'm going to write him on here. Probably should have used black. Okay, ATP synthase. All right, so these are all of our players. You do not need to know these individual proteins' names, okay? All right, so step 1 is going to happen over here, all right? Step 1 is where our light energy is going to come in, and it is going to be absorbed by our chlorophyll. Okay, so chlorophyll, it's not actually what these circles are representing, but we're just going to kind of keep it easy for right now. Okay, so that is step 1. Step 2 is going to happen down here in the lumen. Okay, so in the space inside, we're going to take 2 water molecules. Okay, and the water molecules are going to be broken by aspects of the photosystem. Okay, and it is going to be broken into 4 hydrogen ions and 2 oxygens. That oxygen will then just poof, not poof, but it will be able to leave the plant. All right, but because we have positive ions right here, that means that's where our electrons went, okay? So this light energy is going to travel through, and it is going to join with 2 chlorophyll molecules that are going to be here. And when the electrons and the chlorophyll molecules interact, the chlorophylls can excite those electrons. Okay. So now that was step 2, the photolysis. So the photolysis, using the light energy from the sun here, we're splitting that water. We're having a buildup of hydrogen ions. We're able to release the oxygen. All right, so step 3, the electrons travel from photosystem 2 to photosystem 1. So step 3 is the journey of the electrons from photosystem 2 to photosystem 1. Okay, these little guys actually move or shuttle, but what's happening as we go through this one, remember, I also mentioned that we have hydrogen ions that get pumped into our lumen from our stroma space. All right, so our electrons have gone on our long journey from photosystem 2 to photosystem 1. So again, we've got some chlorophyll molecules. Okay, chlorophyll molecules. Here, I'll label them on this one, chlorophyll molecules. Chlorophyll molecules, okay? So just like before, for step 4, our light energy from the sun, it's going to come in, it's going to move down and around, and the chlorophyll will pass the energy to the electrons. All right, so now step 5. Step 5 is going to be kind of over here. These electrons are going to get passed to this guy here, and he is going to take NADP plus H plus, and these 2 electrons, because there's 2 of them, and he is going to form them into NADPH. So that's our electron carrier. So notice that the NADPH is in the stroma. Okay, he's in the stroma. All right, so that is step 5. And then our last guy, so we have the 4 hydrogens here. We have pumped another hydrogen in. This is happening repeatedly, so we have lots and lots and lots of hydrogen ions in our lumen. We don't like to have a buildup of hydrogen ions, so that's when we're going to have step 6. Okay, step 6, the ATP synthase is going to be activated by the buildup of hydrogen ions in the lumen, and it will pump those hydrogen ions back out into the stroma. When it does that, that is going to cause this part down here to be able to rotate. And as it rotates, ADP plus a free phosphate ion are going to be joined together to form ATP. ATP is also in the stroma. So if you go back and up here to our products, we've made O2, and even though we made that in the lumen, it's going to be released by the plant. But the NADPH and the ATP were made in the stroma, NADPH and ATP made in the stroma, and that's really important for our next step. All right, so for our next step, oh no, my words are upside down. Oh, fiddlesticks. Well, I will have to just fix that, okay? So I'm going to pause right now to write what I need to write. All right, so I fixed my words. You can kind of see, don't know why I printed, I do know why I printed up there, but I made an error. So we're going to have reactants for this step is going to be carbon dioxide, NADPH, and ATP, and our products are glucose. That's really the only product we care about. There are 3 steps to the light independent reaction, or the Calvin cycle. The first step is carbon fixation. This is really important aspect of photosynthesis. An enzyme called rubisco combines carbon dioxide gas and 3 existing 5-carbon molecules, they're called RUBP, you don't need to remember their name though, into 6 3-carbon molecules. So that's the first thing, is that we're taking gas and combining it with a solid molecule into a permanent solid molecule. Then in step 2, we have reduction. So remember oil rig, oil oxidation is losing, rig reduction is gaining. So the carbon molecules that we just made from carbon fixation are going to be reduced by NADPH. Remember that NADPH is an electron carrier. So what does this mean? They're reduced, so the carbon molecules get the electrons they're gaining from the NADPH. And then in step 3 is regeneration. Just like you would think of regeneration anywhere else, we are going to release the 1 3-carbon molecule that we've made, but then we're going to get the starting molecule, RUBP, reformed using the last of the energy from ATP. It is important to note that to make 1 glucose molecule, the cycle must be repeated. So notice in regeneration, we're just making 1 3-carbon molecule. So we've got to do this twice to get 1 glucose. All right, let's annotate our picture now. All right, so here you have carbon fixation, which is the first step, reduction, which is the second step, and regeneration, which is the third step. So here we have this, all of these dots or shaded circles are going to be our carbons. So this is 3 CO2 molecules, okay? So remember that this is gas. This is really important that we remember that this is a gas, okay? So we're going to have 3 of these individual carbon molecules, okay? This guy over here, we have 3 of these, 1, 2, 3, 4, 5 carbon molecules. This is our RUBP, so you don't need to know that, but it can be helpful. And Rubisco is our enzyme. So Rubisco, really should be over here, huh? Rubisco is going to take these 2 guys together and combine them to make this 1, 2, 3, 4, 5, 6 carbon guy. Okay, let's do a little carbon math real quick. Okay, so this guy here, if we have 5 carbons on him and we have 3 of them, then that means that we have how many carbons total? We have 15 carbons total. And then if we have 3 CO2s or 3 carbons here, okay, if we add 3 to 15, what do we get? Okay, we get 18, right? So let's look. We have 3 of these 6 carbon guys. Do we have 18? Yes, we do. We have 18 carbons total. Now, this dude here, he is a solid. Very important to remember, but he is extremely unstable. Very unstable. So very quickly, he is going to go from these 3 6-carbon guys into 6 3-carbon guys. It's not quite as easy as just splitting in half, although that's what it looks like, right? But do we still have 18 carbons? Well, if we've got 6 groups of 3, yes, we're still going to have 18 carbons total. So it's really important that we do our carbon math. Okay, so now we've got this guy. This guy has to go through reduction, okay? Remember, we're only focusing on the carbons. We have 6. It looks like we have 6 here, but we need to transfer some electrons and reduce this molecule. So the first thing we're going to use is 6 ATP. Where did we get our ATP from? ATP were from the light dependent reaction. Because where is all of this taking place? All of this is taking place in the stroma. So we were in the stroma when we produced the ATP, so it's right there ready to go. So we are going to get 6 ADP. These guys will be able to go into the light dependent reaction to form more ATP. But even though we've released some energy, we haven't released any electrons. So now we need to use our NADPH, okay? And we're going to form NADP+. So this is our reduction step. That's where our electrons are transferred. So now we're going to release one guy. This one guy is 3 carbons total, right? Well, we have to do 2 cycles times 2. Got to do another cycle, and then we're going to get to equal 6 carbons total. Oh no, my purple pen. There we go. Which is our glucose. Okay, so we have our 5 molecules of 1, 2, 3. We've released 3, so this is 15 carbons total. We know that we are going to end up with 15 carbons total here, so we just need to get them, instead of being in 5 groups of 3, we want 3 groups of 5. So we're going to use our ATP and then form a DP over here, okay? All right, so I'm just going to make one more note over here. This NADPH is also from the light-dependent reaction. All right, so we're going to spend some time in class going over this and processing this, but hopefully you found it a lot easier than cellular respiration. Let me know what questions you have when you get to class.